And just to confirm, everyone can see me and everyone can hear me?
All good.
Oh, good. Excellent. Okay, so I'm Dr. Rebecca McHugh. I am a postdoc in Paul Hoskison's lab at the University of Strathclyde. And firstly, thank you to the organisers for giving me a chance to kind of keep involved with the MRF AMR scheme. For those of you who don't know, back in 2018, I was lucky enough to do the kind of residential course in Bristol. And it really was a great experience. So I hope you all get a chance at some point over the next couple of years, hopefully to let you do that. So I've titled my talk today, Exploiting Streptomyces Natural Products to Combat Difficult to Treat Infections. And I know that phrase might sound a little bit odd, but hopefully it will become clear to you why I've used difficult to treat rather than just resistant infections over the period of time that I'm talking. So firstly, I'm going to go through an introduction basically to streptomyces. Is everything okay? Yep. Is everything all right?
Everything's fine.
Okay.
Everything's
That's
fine.
fine.
Yeah.
Sorry, I can just hear someone else in the box. So firstly, I'm going to talk to you all today about a bit of and give you a bit of an introduction to streptomyces natural products and their use and their relevance in our search for new antibiotics. And I'll then give a little bit of an overview of the work that I carried out during my PhD. And then I'll talk to you a little bit about the work that I've done over this short and kind of limited by COVID postdoc. And then I'm going to try and play a game with you, but I might be stretching the technology a little bit at that point, but I'll give you the link to that all at the end. So hopefully that suits. Next slide. I'm just trying to get the slide to change. There we go. So streptomyces are these multicellular soil-dwelling actinobacteria, and in terms of medicinal molecules, over the years they've been a rich source of those. So streptomyces, you can see on the left, they have this unique and fairly complex life cycle, starting out with a spore, which will go on to form vegetative hyphae, then aerial hyphae, and then this aerial hyphae will divide into spores. And it's at this point, at the aerial hyphae and spore formation, that we start to see the induction of the expression of a whole host of interesting natural products. These can be antibacterial products, antifungals, anti-cancer, even immunosuppressants, and some pigments that are of particular interest to us. And this makes them a kind of good place to start looking when we want to find new natural products, or and specifically at this point in time, new antibiotics to try to combat antimicrobial resistance. So you can see their morphology and their phenotype can look pretty different across the species and across the different media. And again, we can get a real diverse bunch of molecules produced by these organisms. So in our top left and our top right, these were are strains by John Munnock in our lab, and they're Streptomyces silicolor, or Streptomyces model organism, and the blue pigment that they're producing is actinorodin, a kind of weak antibiotic produced by Streptomyces silicolor, and it's this lovely blue pigment. In the middle, you can see Streptomyces goldeniensis grown in a whole lot of different media. And you can see how the phenotype changes, and you'll hear a lot about Streptomyces goldeniensis throughout my talk, as it's the producer of Orodox, the main natural product that I worked on throughout my PhD. On the bottom left, we've got Streptomyces claviligirus. This is the producer of clavulanic acid, which is a beta-lactamase inhibitor. And it's often used to try and combat AMR. This is the natural product that I'm working on for my postdoc project, so I'll be talking about that a little bit to you today. We've got some other interesting Streptomyces species there. You can see how different they all look. We've got Avermetilus, the producer of ivermectin, and Streptomyces venezuelae, the producer of chloramphenicol, although that's synthetically produced now. It was originally discovered in Streptomyces. So I hope that kind of recaps how important these molecules have been, these organisms have been in terms of our molecule discovery over the years. And if possible, could you please play the video, Daniela? I've got a little video, and this was produced in our lab, by our lab, and also a stop motion animator collaborator that we have, Anand. So hopefully we can play that for you just now. And this gives you a little bit of an introduction about this, about streptomyces.
Okay, can you see that video?
I can see that, yep.
Okay, there you are then. There you are.
You know that fresh, earthy scent that you can smell just after it's rained? It's thanks to a group of bacteria called streptomyces. In the soil, these microbes produce a molecule called geosmin, which fills the air with its distinctive musty odour when raindrops hit the ground. But there's a lot more to streptomyces than their smell. These bacteria recycle organic matter by feeding on dead organisms. And they help to buffer our soils, stopping them becoming too acidic or alkaline. But one of the most important things that streptomyces do is produce antibiotics. The vast majority of streptomyces bacteria don't actually make us sick, but they produce over 2/3 of all known antibiotic compounds. We use antibiotics every day to fight disease and improve the growth of livestock. But why do bacteria produce them in the first place? They could be used as a kind of chemical warfare against other microbes. But they may also act as communication signals, change how genes are expressed, or modify the way bacteria colonize surfaces in 3D structures called biofilms. Whatever their natural function, we're able to utilize the amazing properties of these molecules as medicines to treat bacterial infections. But some bacteria have evolved resistance to antibiotics. These include superbugs like MRSA and other bacteria which can be almost impossible to treat. Antibiotic resistance is one of the greatest threats we face today. By 2050, it could kill 10 million people every year. That's more than current deaths from cancer. One way to prevent the problem is to reduce our use of antibiotics in healthcare and in agriculture. But we also need to find new kinds of antibiotics, and streptomyces bacteria could be just the place to look. They are an incredibly successful group of organisms found almost everywhere on Earth. And scientists are searching the deep sea, tropical forests, and deserts for undiscovered species that may produce new kinds of antibiotics. We may also find novel antibiotics by looking at the DNA of streptomyces bacteria. Some species have the capability to produce dozens of antibiotic compounds encoded in their genome, even though they only produce 1 or 2 of these in the lab. If we could trigger the expression of these silent genes, they could be a vast new source of antibiotics. Streptomyces have been producing antibiotics for millions of years, long before humans even existed on this planet. Now, our future here may depend on them.
Thank you, Danielle.
That's all right. Let's stop sharing that.
I'll share this PowerPoint slideshow again. So can everyone see this?
Yeah.
Yep.
Yeah, great. So hopefully that gave you a little bit of an overview of the importance of these organisms with some nice kind of cute cartoons to go with it. And that kind of summarises their importance, but obviously I'll get down to a little bit more of the detail with you this afternoon in terms of what we do in the lab and my project and how it relates to this. So as was mentioned in the video, streptomyces natural product isolation has traditionally taken place through isolation of soil organisms. So this involves taking a sample from anywhere really. The video mentioned deserts or tropical forests or marine environments, and you can take that soil, bring it back to the lab, and you can isolate bacterial strains from this soil sample. And once you can, as the second image there, you can see in this strain isolation plate, you start to see a diverse group of molecules, and you can select streptomyces or other actinobacteria that you think might have antibiotic producing potential. And at this point, you can streak these strains onto different media to try and induce the production of some of these gene clusters, or induce the expression of some of these gene clusters that were mentioned in the video that might be silent otherwise. And then you can either take the agar that they've grown on directly, or you can grow them in liquid media and extract compounds to carry out bioassays that you can see on that plate furthest to the right. And it's at this point that you would like to be able to carry out some analysis, perhaps LCMS or NMR, to try and identify a peak that corresponds to the antibiotic that you've been looking at. So you might be able to get the structure of that through NMR, for example. And that's great if you find a new antibiotic that way, fantastic. But there obviously are issues with this traditional method. So there are advantages, obviously. Many of our traditional antibiotics were discovered via this method. So if you think about streptomycin, Waxman and Schatz, they isolated Streptomyces griseus, the streptomycin producer, from the soil, and they went through this method and found the antibiotic. However, rediscovery is an issue. So you could go to the Atacama Desert, or you could go to the Borneo rainforest, or you could go to the bottom of the ocean, and you could go through that whole process, and you could get to the end, and you could still find, and you could think you've got a great antibiotic that works against all the pathogens you've tested against, and then you find at the end that it's streptomycin or erythromycin or something similar, a class of antibiotics. So it tends to be that even in the ecological niches, we can still see a lot of overlap between the biosynthetic gene clusters across the world. They're quite shared. So we do get rediscovery quite commonly, and it's a lot of effort to get to that point to rediscover a molecule that's already in the clinic or has been discovered before. We've also got the issue of this great plate count anomaly. So metagenomic analysis tells us that in each gram of soil, we've got millions or billions of bacteria. But when we actually take that bacteria and plate it out in the lab, we don't see as many. We can sometimes only get as few as a few hundred colonies, or even less than that. And that's because less than 1% of bacteria in soil are actually culturable in the lab. And there's been some new techniques to try and get around this. So you can see here the iChip. So the inventors of the iChip wanted to try and get around this issue by immersing the chip in the soil to give the bacteria that they'd isolated the exact nutrients that they would have in their environment. And this new method actually discovered a new species of bacteria, this Eleuthera terri, that produces a novel antibiotic, teixobactin, that's actually kind of making its way through the clinical development stages at the moment. So there is still potential out there. It's just we have to find new ways of unlocking it. In terms of novel technologies, next generation sequencing has been really important for natural discovery, natural product discovery, and especially in streptomyces. So if you cast your mind back to 2002, maybe some of you were very young at that point, including me, pre-COVID times. This is when the genome of the model Streptomyces organism, Streptomyces silicolor, was published, and that was quite a surprising announcement to all of us, because everyone expected that Streptomyces silicolor would have gene clusters that encoded the natural products that we already knew about. So the actinorhodin cluster and decel prodigiosin cluster. No one expected there to be more than 20 genomic regions encoding for specialised metabolites, because we'd never seen their production in the lab. So essentially, this was the realisation that many Streptomyces species might have silent biosynthetic gene clusters that are unable to be produced in the lab and encoded within their DNA. And so then the question was, can we try and unlock some of these natural products and try and either express them within their native strain, or can we take the DNA and express it in a kind of another superhost strain in order to actually analyse that particular natural product and assess its antimicrobial potential. So this is typically done by sequencing a whole genome of an organism. So this is my pet organism here, Streptomyces goldeniensis. And you can run a software program called Anti-Smash, and it will literally just spit out a list of all the different biosynthetic gene clusters that are in there, what they're similar to, and what the software program expects it to encode. So we see lots of natural products like terpenes that are common among the Streptomyces species across the planet, and stuff like geosmin, that smell that was discussed in the video that you can often smell after it's rained, but also some natural products that are quite actually specific and individual to each strain, and those are the ones that we're kind of interested in. And actually, there's several examples of how we can use this combination of exploiting new niches as well as genome sequencing to try to identify new natural products. So Dr. Rebecca Devine, who works in Professor Matt Hutchings' group at the John Innes Centre, who was also on the 2018 course with me, works on Streptomyces that have been isolated from these fungal farming ants, or Tetraponera. So these ants actually carry Streptomyces on their chest, and these Streptomyces actually work to try and produce natural products that actually protect the fungal farm that these ants actually make. So Rebecca and everyone who's worked on the project have been able to isolate these bacteria and sequence the whole genome, and discovered a few interesting natural products that are produced by these Streptomyces strains. Over 35 biosynthetic gene clusters were encoded, and they found one particular compound or group of compounds of interest. These are the formicomycins, and these are active against VRE and MRSA. So this just shows the kind of validates the kind of process of genome mining and exploiting these new ecological niches for the identification of novel strains. Another kind of technique that we might have is the repurposing of traditional or shelved antibiotics. So you've probably all heard, or you've definitely seen this figure on the left-hand side, and this whole concept of the golden era of antibiotic discovery, where many antibiotics were discovered over a fairly short period of time. And in the last 20 years, we've not really seen many antibiotics or new classes of antibiotics be discovered or get their way onto the market. But while all these amazing antibiotics were being produced, the standard was very high. So some antibiotics that might actually still have use to us today or have other purposes were kind of shelved while the pharmaceutical companies were investing a lot of money in antibiotic discovery at that time, and they were isolating thousands and thousands of compounds at a time. Very many of them were selected out fairly quickly. So a lot of my work is involved in looking at some of these projects and determining whether or not we can repurpose these molecules for novel methods of treating infection. And my project is focused on the antivirulence concept. So antivirulence treatments differ from traditional antibiotics, as we are not aiming to try and actually kill or wipe out or severely inhibit the metabolic processes of that bacteria. We're simply trying to prevent those bacteria from being able to infect the host to the point where the host is severely damaged. So in order to do this, you have to have a good target, and obviously different bacteria or different infecting bacteria have different targets that we can use to design or to use antivirulence molecules, and this is what my PhD project is focused on. And it's been focused around this molecule here, Orodox. So Orodox was discovered from Streptomyces goldeniensis. This is a strain of Streptomyces discovered in Bermuda back in the 1960s. And it was originally studied for its ability to inhibit the growth of gram-positive organisms. However, it didn't make it to the clinic. Structurally, you can see it's quite similar to chiromycin from Streptomyces calinus. The main difference being the methylation to the pyrrole ring that I've highlighted here. Chiromycin doesn't have that methyl group, which we think is actually fairly important for its activity. As many of our shelved antibiotics were, it was used as a growth promoter in poultry for a while, but it never actually made it to the clinic in terms of its an antibiotic. So it was well studied at the time. We know its mechanism of action. We know that Orodox binds to elongation factor thermal unstable, or EFTU. So this protein is responsible for delivering tRNA to the ribosome during protein synthesis. So typically EFTU will gather tRNA, deliver it to the ribosome, and then a GTP hydrolysis will take place, releasing EFTU back into the protein synthesis cycle, and the cycle will continue. However, when Orodox binds, although EFTU can still take the RNA to the ribosome, this GTP hydrolysis is blocked, meaning that EFTU can't dissociate from the ribosome, and the protein synthesis cycle is blocked, and therefore the protein synthesis cycle will cease and the cell will die. What's of particular interest to us that in 2011, Orodox was pulled out of a very large-scale natural compound screen as a potential inhibitor of the type 3 secretion system in enteropathogenic E. coli. So why is this important? Well, we need novel treatments for enteropathogenic and enterohemorrhagic E. coli. I feel like I have a bit of a cheek saying this during COVID, but they are significant global pathogens. And in Scotland, we have a particular issue with EHEC, E. coli 0157, where we tend to see around 3 times as many cases as the rest of the UK. And it's particularly harmful in children because enterohemorrhagic E. coli encode these sugar toxins. So that can actually cause damage out with the gut, and in particular to the kidneys and the brain. And it can cause this hemolytic uremic syndrome, or HUS, and that can be fatal, especially in children. And these bacteria use this type 3 secretion system for their host cell attachment. So the idea would be, if we could block this system, the E. coli wouldn't be able to colonise the gut during an infection process, and therefore wouldn't be able to cause that significant amount of damage. We've also got issues with typical antimicrobial resistance in enteropathogenic E. coli, and therefore antivirulence treatments might be a solution to that as well. So it's important to note that currently antibiotic or traditional antibiotic treatments aren't recommended for enterohemorrhagic E. coli infections. That's for 2 reasons. The disruption caused by these antibiotics to the gut microbiota can actually have negative consequences and cause an increased severity of infection. But most importantly, these traditional antibiotics induce a bacterial SOS response in EHEC, and that SOS response actually leads to the upregulation of sugar toxin production, meaning that it's more likely that the infection will shift to the gut and cause the damage in the kidneys and other areas of the body. So we need to try and find novel treatments and novel concepts to treat E. coli infections, or these E. coli infections of the gut. So can we use Orodox as an antivirulence treatment? We know that it has this potential type 3 secretion inhibitory property. So the first kind of aim of my PhD was to try to determine the effect of Orodox on type 3 secretion. So I've got EPEC, EHEC and Citrobacter rodentium here, which is the mouse model pathogen for these infections. And you can see that when we add increasing concentrations of Orodox to our samples there, we see decreasing quantities of type 3 secretion system associated proteins in the supernatant, until we get to about 5 micrograms per mil, where we don't see any type 3 associated proteins whatsoever. It's important to know that at this highest concentration, 5 micrograms per mil, we don't see any effect of Orodox on the growth of the bacteria on any of the strains, suggesting to us that the mechanism of action of Orodox might be or this type 3 inhibition by Orodox might be a separate mechanism from the EFTU binding that we know happens in gram-positive bacteria. And then to check in a cellular model the effect of the compound, we set up this HeLa epithelial cell model that we infected with GFP tagged EHEC bacteria. So on the top, you can see our uninfected panel where we just checked that the compound itself didn't have any negative effects on the cells, which it doesn't seem to. And then on the bottom left, we've infected these cells with EHEC and left them untreated. And you can see, even just looking at the size and the shape of the cells are being very much harmed by these EHEC bacteria. And if you look at the zoomed in panel here, you can see that the EHEC bacteria are starting to form these microcolonies on the surface of the cell. And you can see the actin start to condense and form pedestals, which is really characteristic of an EHEC infection. And again, the cells are even starting to round, and we can see cell death in some examples. On the right-hand side, when we've treated these cells with Aurodox, although the bacteria seem to be existing there fairly happily, they aren't able to actually form these tight junctions with the cells and significantly form these actin pedestals that are so characteristic of EHEC infections. And the cells themselves, they look much healthier. And when we actually quantify this, we see a threefold reduction in the number of cells infected. And we see a threefold reduction in the number of cells infected, and actually a 7,000-fold reduction in the colonisation efficiency of EHEC when treated with Aurodox. So it has a significant ability to reduce these EHEC infections in our cellular model. So we had a few kind of ideas of what we thought might be going on. So we carried out, in terms of a mechanism of action, so we carried out some whole transcriptome analysis to see if that could give us any ideas. So you can see that actually only a small number of genes in the genome were actually affected by Aurodox treatment. This is typically a lot less than a traditional antibiotic. But of the genes that were downregulated, or the genes that were affected by Aurodox, we homed in on this Lee pathogenicity island that encodes for the type 3 secretion system. And we found that actually 41 of the 42 genes present in this pathogenicity island that encodes for the type 3 secretion system were downregulated in the presence of Aurodox, suggesting that a transcriptional inhibition of expression may actually be the mechanism of action of the compound. It was of particular interest to us that LAIR, this gene I've highlighted here, which is a master virulence regulator in E. coli, was downregulated significantly in the presence of the compound. And what was also notable was the fact that other genes regulated by LAIR out with this Lee pathogenicity island were also downregulated in the presence of the compound. So these include some biofilm genes, heat shock responses, etc. So it gave us an idea that LAIR could be, or the regulation of LAIR in some way could be the target of Aurodox. So on the left-hand side, we just validated that by a GFP reporter assay, and you can see that in the presence of Aurodox, LAIR expression is significantly downregulated, especially when compared to our housekeeping gene control. And you can see on the right-hand side, when we overexpress LAIR, this type 3 secretion system knockdown phenotype in EHEC is completely reversed, showing that potentially LAIR is the target if extra LAIR or the presence of LAIR can actually reverse this knockdown phenotype. So our theory at the moment is that the LAIR regulatory pathway in some places is the likely target of Aurodox, but then we also have to understand a bit more about the LAIR regulatory pathway to kind of pin that down. And I'm actually going to be starting on an MRC funded grant over at Andy Rowe's lab at the University of Glasgow later on in the year to try and get to the bottom of exactly where Aurodox is targeting within EHEC and EPEC. So Shiga toxin production, as I mentioned at the start, Shiga toxin production is really detrimental to the host, as this is what facilitates the effects on the kidneys and the brains that we see in hemolytic uremic syndrome. So it was really important for the development of this compound that Aurodox didn't induce that like a traditional antibiotic would. So we first of all checked for the expression of the SOS response, again via GFP reported assay, and we can see that Orodox did not induce the bacterial SOS response, especially when compared to ciprofloxacin, which you can see pushes the expressions of these SOS genes through the roof. And then when we blotted for sugar toxin itself, this was done in Citrobacter rodentium because we don't have the license to use sugar toxin producing EHEC within our lab. But you could see that there was actually no production of sugar toxin, or no detection of sugar toxin in our Orodox sample, whereas we could detect it in our ciprofloxacin treated sample. So it was really useful to us that the Orodox didn't induce that sugar toxin response. So we know that Orodox is good at producing, or we know that Orodox is good at inhibiting these the E. coli and its ability to attach to the epithelial cells of the gut. And we know that it doesn't induce sugar toxin production. But what we didn't know at this point was very much about Orodox biosynthesis. So this is where my work on Streptomyces comes in. So we wanted to try and kind of decipher Orodox biosynthesis through the characterization of the biosynthetic gene cluster that encodes for Orodox. So we sequenced the whole genome of Streptomyces goldeniensis and identified the Orodox biosynthetic gene cluster through the software program Anti-SMASH that I mentioned earlier on in the talk. So you can see here that we found this Orodox biosynthetic gene cluster, and it stood out to us as it had a really strong similarity between the Orodox biosynthetic gene cluster and the chiromycin gene cluster that had previously been characterized. One major difference is the gene that I've highlighted in the red box. This is SAM-dependent methyltransferase present in the Orodox cluster that isn't in the chiromycin cluster, which makes perfect sense because Orodox has a methylated pyrrole ring that chiromycin doesn't have. And overall, 23 of the 25 genes in the Orodox cluster had functional homologs in the chiromycin cluster. So we were pretty sure that we'd found the right gene cluster at that point. But to characterise this methyltransferase, I cloned it and expressed it in the chiromycin producing strain. And then we managed to get conversion of chiromycin to Orodox, showing that the methylation of Orodox is the last step in its biosynthesis, and that it's catalysed by this ORM star that we have in the Orodox biosynthetic gene cluster. So we have a fairly good idea how Orodox is actually produced, or we had a fairly good idea of how Orodox could be produced in Streptomyces goldeniensis. But to confirm that this actually was the gene cluster that encoded for Orodox, we cloned and expressed the Orodox gene cluster onto a back, and that back was then expressed in our Streptomyces coelocolour superhost M1152. And then you can see here that we start to get this Orodox associated peak and this Orodox associated peak when we express that back in Streptomyces coelocolour, and we could confirm that by LCM, by our MS analysis and by bioassay, the Orodox was present there. So this was the gene cluster that encoded Orodox. I also have knocked out the polyketide synthase, or the essential polyketide synthase genes from the Orodox gene cluster, and we see almost abolition of the Orodox production. We start to see kind of Orodox derived smaller molecules when we knock that gene out. So that really pins down that we do have the right cluster. It's also of interest to us, or it was really satisfying after working on this so long, that actually when you express Orodox in Streptomyces coelocolour, you can see it grows bright gold, which is the colour of the Orodox compound itself. So yeah, that was quite nice to see. Elongation factor 2, as I mentioned at the start, is an Orodox target in gram-positive bacteria. So many species that produce these Orodox-like molecules also carry an extra copy of elongation factor 2 that is resistant to these alpha-mycin or Orodox-type compounds. So our question, and when we tried to express the Orodox back and other strains that didn't have this extra copy of elongation factor 2, we didn't see much orthodox biosynthesis. So we wanted to determine whether or not that extra copy or that resistant copy of EFTU was in fact needed for orthodox biosynthesis. So in Streptomyces venezuelae, I expressed an extra copy of the elongation factor 2, and we started to see very large quantities of orthodox produced once we had the BAC and this gene in place. It's showing us that both genes are required for orthodox immunity during biosynthesis. So that was of particular interest to us. So just in terms of the orthodox work, in summary, orthodox inhibits the attachment and effacement of EHEC to epithelial cells via the downregulation of the master virulence regulator layer. And I've identified the orthodox biosynthetic gene cluster and some of the resistance mechanisms. So our future work will involve some mutagenesis studies and some biochemical analysis to try and make ultimately try and make some orthodox derivatives and test them in the system. And that will be done hopefully by myself and Robin, our PhD student that's joined the orthodox project. So as I say, I'll be going back to work on that towards the end of the year. So just in summary, a little bit about my postdoc project. I started this project last summer, so obviously I've been a bit inhibited by COVID, and I also can't actually tell you too much about it because of the fact that it's in collaboration with GSK and there's a lot of IP involved. But essentially, this project is based on clavulanic acid. So clavulanic acid is this beta-lactamase inhibitor. You can see it, the structure of it there. So it will competitively inhibit the beta-lactamase enzymes compared to when it's prescribed alongside something like amoxicillin and this augmentin that you can see here, that's the GSK brand name, and therefore allow that to kind of work in the way it should. So it's produced by Streptomyces claviligirus in traditional large-scale fermentations, and that happens at Irvine, which is just south of Glasgow. So we have a collaboration set up between ourselves and GSK to try and improve this process. So first and foremost, these strains that I'm working on at the moment are actually early production strains. GSK have another production strain that I'm not allowed anywhere near, I'm allowed to get my hands on at all, that they actually use in their fermenters. And that strain is very, very good at producing clavulanic acid. And it's been mutated over time through various, again, mechanisms that I'm not allowed to know. It's been mutated over time in order to produce this very high clavulanic acid titer. But over time, this mutagenic process has meant that the stresses have caused a reduction in the ability to use different carbon sources. So they've lost their metabolic flexibility. And this is a phenomena that occurs across many industrial Streptomyces lineages. We see that as the titer of the product that we want goes up, the metabolic flexibility is reduced. So that's obviously a problem because they're now very adapted to the fermentation media that they use. And if we want to be more sustainable and try and use feedstocks that come from example, waste products from other industries, then it can be a bit of a problem. It can be a bit of a problem because these strains have lost their ability to use a lot of these complex sugars. So my project is kind of revolved around trying to make sure that Streptomyces claviligirus can try and use some of these carbon sources that had previously lost the ability to use. So we, or with the help of John Munnock, or another postdoc in the lab, the industrial strain lineage was sequenced, and we identified mutations in these genes that could lead to this reduction in metabolic flexibility. And I'm just going to give you one example today. So this is GLPX. We found a stop codon about 1/3 of the way through the gene. So we know that our later strains or our later industrial strains didn't have a functional GLPX gene, and it didn't have a functional fructose 1,6 biphosphatase. And this actually meant it probably led to higher clavulanic acid titers, because if you can't do the reaction between fructose 1,6-biphosphate to make fructose 6-phosphate, all that fructose 1,6-biphosphate is going to make glyceraldehyde 3-phosphate, which is a direct clavulanic acid precursor. But also it might reduce the number of, it might inhibit a number of different carbon sources being used in gluconeogenesis. So we carried out some analysis using a biolog system, which is a phenotypic array where we look at the ability of the strains to use different carbon sources. So on the left, I've got an early GSK production strain, SC2, versus a later GSK production strain, SC6. And you can see that, and anything towards the kind of colour purple is indicating that SC6 can't use that carbon source as well as SC2 can. So we see a lot of purple. On the left-hand side. When we then restore the functionality of GLPX back into SC6, we see everything's much more pink, or much more in the middle, meaning that the ability to use these carbon sources is actually much more comparable. So this has obviously got to be, this is in very small volumes in a 96 well plate at the moment. So the next kind of work on that will be trying to validate this in a more fermentation-like setting, and working on a whole host of other genes that I've also identified that could potentially have effects on how the carbon is used. Also, surprisingly, when I added GLPX back into SC6, we actually saw an increase in clavulanic acid titer, and also we saw an increase in biomass, which makes sense if we can use a lot more energy sources. So again, validation will be important over the next few months, and as I finish off this postdoc. So in conclusion, the whole genome sequence of these industrial strains can be identified, used to identify targets for metabolic engineering, and the restoration of GLPX improves metabolic robustness in Streptomyces claviligidis in our GSK industrial lineages. So as I say, my future work will involve the validation of other targets, but also I'm working on a system to speed up the mutation process in Streptomyces. So if any of you have worked with Streptomyces before, you'll know it can take a long time to make mutants. This is partially due to the fact that Streptomyces won't take up methylated DNA. So we have to pass everything through non-methylated E. coli in order to do that, and then use conjugation. So I'm currently working on a suite of Streptomyces vectors that do not have any methylation sites. So we can kind of bypass this and speed up the process. So that's what I'm going to be focusing on until I move back over to Andy's lab towards the end of the year. And just quickly, one other thing that we've been working on as a lab is this game, Dr. Dirt. So it's a kind of simulation of how we identify new antibiotics in the traditional method from the ocean or from the sea. So I would encourage you all, if you want to, you can play it on this website, or it is actually an app in the App Store. It's a free app, and it's really good. We found anyway for kind of public engagement and stuff. We launched it during Science Week and we got really good feedback from it. So essentially you can drag a test tube around the different environments. So I've just chosen the kind of marine environment for you at the moment, but there is a terrestrial environment as well, and it will give you information as you do this. I didn't want to show you the actual game because again, technology is a bit limited. So we'll drag this all around, and you'll get some information about the different bacteria that are associated with the different areas. And then once you've done that, you can take your test tube back to the lab and you can isolate the bacteria from it, and then you can drag and drop these bacteria into the culture box, and then you can do some bioassays and see if the bacteria are making anything that kills the indicator organism. So you can try with staph or E. coli or candida, and then you can then inject your extract into this LCMS machine. and you can see whether or not you actually got an antibiotic compound. And if you play on the App Store, you can actually collect all the different antibiotics that you can find. So as I say, it's quite a good tool for public engagement alongside the video that I showed you earlier. So I think in the interest of time, I'll kind of wrap up and give time for questions, but just I would like to say thanks to everyone at the University of Strathclyde and Paul Hoskisson's group. John Munnock's helped a lot with the genome sequencing and things like that. The whole of the Strathclyde microgroup, and at the University of Glasgow, everyone that was involved in that Orodox mechanism paper that we got out a couple of years ago. And the game doctor who helped us design the game, Dr. Carla Brown, she's fantastic and she's helped us a lot, or helped us get that game out there. And then obviously this AMR funded cohort that has brought me here today, and I'd be happy to take any questions.
Thank you, Rebecca. Thank you for a really, really wonderful talk. If anyone does, thank you. if anyone does have any questions, please feel free to write them into the chat function. And we'll just wait a few minutes before we get some questions. In the meantime, whilst we are waiting, I had a few questions myself. Unless anyone wants to speak up now, feel free. Yeah, Ryan, we'll take a question from you.
Hello. Thank
Hi.
you. That was a fantastic seminar. As somebody whose work just points out problems, it's always nice to hear from someone who's actively trying to find solutions. It was really nice as well hearing how you've carried the work from your PhD into your postdoc and moving forwards. My question is, and sorry, it's a really negative question, really pessimistic one. Do you think that with the discovery of new antimicrobial compounds, it could just be putting a short-term plaster on a much deeper wound?
Oh, a philosophical question. I mean, yeah. There's all there. We got into actually quite a lot of deep discussions about these kind of talks when we were in Bristol, and I think that you could definitely look at it that way. But I think a lot of the issues that stemmed from the overuse of antibiotics, or that we have at the moment, obviously stem from the overuse of antibiotics when people tended to treat them like they were unlimited and they were going to go on forever, despite Fleming warning us in his Nobel Prize speech that if we used it without thought, that we would start to see resistant organisms. He could tell back then that was going to happen. So I think now that we're much more aware, we can use these compounds much more sparingly. But yeah, I think we definitely need to look for new concepts as well as new compounds that are going to help us with the crisis that we have kind of looming at the moment. So that's up to your cohort to try and do that.
Yeah, sorry for putting a downer on it. Good answer there. And so another thing I thought that was quite interesting, when you were comparing your gene cluster with a similar cluster,
Yeah.
obviously they were highly similar, but some of the orientation was the other way around, which suggests to me that maybe there's been some recombination events with these clusters being formed. Do you know if anyone has looked into how these clusters originate?
Yeah, me. So I didn't actually get into it too much today, but in my thesis and hopefully in a paper that we're going to get out soonish, we've looked into how these clusters potentially arose and the origin of that methyltransferase. Did it come from another organism? Did it come from somewhere else within the genome of Streptomyces goldeniensis? And we've actually used some recombination detection software programs to detect recombination events between the Orodox and the Chiromycin cluster. And also, what I didn't really mention today is that actually the Orodox cluster is the Orodox encoding region is actually part of a much larger supercluster that's around 300 kilobases. So we see a lot of recombination between the Chiromycin gene cluster and these other gene clusters that flank the Orodox region. And at the moment, we're trying to sequence some other organisms that produce similar compounds to try and get a better idea of how that recombination might have occurred. But yeah, so we're kind of working on it at the moment. We're literally today sent some genomes off to be sequenced. So yeah, hopefully we'll be able to answer that in the next year or so.
Oh, that's fab. I look forward to reading the paper when it comes out.
Fingers crossed.
All right, thanks Ryan, and thanks Rebecca. We've got a question here from Anne Amulele, who says, is Orodox an approved drug or molecule?
It is not an approved drug or molecule, however, and it's very hard to access these historic records of what point the Orodox actually got to in the clinical trial process. But we know that it's had phase 1, we know that it has been in humans at some point, and the problem was the efficacy, rather than in terms of it didn't kill gram positives very well in humans, rather than it wasn't toxic or anything. And you can actually pull the data for that. So we know that it has been in, or it is reasonably safe in humans, but to access that data is actually quite difficult. So we're hoping that it shouldn't be too difficult to get it to the next stage. But no, it's not approved for use in humans at the moment.
As a follow-on question from that, when were these trials conducted? Because obviously now the situation has changed so that we need new antibiotics and everything else. So is that an avenue for readers?
I think it was the 80s. So my supervisor knows everything that we've had in terms of this is kind of word of mouth as well. But yeah, I think it was back in the 80s. Again, information that I'm not allowed to know.
Oh, right. A little bit of Chinese whispers going on then.
Yeah, Chinese whispers.
Arnold Van Villay says, one of the problems with many antibiotics is that they work great in vitro, but when used in vivo, they do not build up a working concentration in the organs or body, or are toxic or nephrotoxic. And that's even before the AMR problem kicks in. Do you have any thoughts on how that can be addressed?
Honestly, it's not really something I've studied over the years. I think most of my work tends to be in the kind of fundamental discovery part of antibiotics. But again, we are focusing, especially with Orodox, now that we know how it's biosynthesized, to try to make a whole suite of compounds that are similar to Orodox, and to try and make derivatives of the compounds so that we can test a whole number at least to try and find one that has a good enough activity and suitable properties in humans as well. So that's the only way that I can, in terms of our compound, that I can think of, is to try and make these derivatives. But it's a tricky problem that we're going to face, I think.
Yeah, indeed, I completely agree. I think this will probably be the final question, unless anyone has any burning questions. So just thinking about this in relation to your postdoc work on bacteria and their adaptation to fermentation media, like in industrial strains.
Yeah.
How much bacterial culturing problem is due to researchers' reliance on historical culturing practice, and should research focus more on discovering different culturing techniques?
Oh, absolutely. And I think the iChip is a really good example of that. And again, metagenomics are coming into it as well. People are finding the genetic components of biosynthetic gene clusters, either having them synthesized or cloning them and expressing them in other organisms that we can culture. So as time goes on, we are finding new methods to do it. So hopefully at one point, I mean, that iChip is fantastic. We've actually had them in the lab before. So hopefully we can come up with more techniques and more culture methods to try and combat this problem. But for now, I think it's still something that we're going to have to cross when we try and isolate these new bacteria in general.
Yeah, it's interesting that you mentioned that, just in terms of one of the reasons I find Sir Alexander Fleming so endearing is that if we think about how he actually discovered penicillin, it was because he went on holiday and did up. So even when he discovered lysozyme, it's like a drop of his nasal mucus fell onto one of the plates he just cultured, and he just thought, there you are. Just, do you know what I'm saying? So quite an unconventional scientist in terms of practice and rigidity to stick into practice and practical techniques. But yeah, it's just an interesting concept to think about kind of in future. Ryan, you've got your hand up.
Yeah, sorry, I just what you were saying then was really, really cool. Yeah, the idea that because a lot of soil organisms are quite hard to culture, you don't actually need to culture them anyway. If you get the sequence and then you clone the putative products, you can then basically do some functional metagenomics, which is really cool. Do you think that will be the future of this kind of work?
I have definitely seen a talk by someone that has done this. So it's definitely a way forward. I'm not sure if the paper's out, but I can try and find that for you. But yeah, there's
you circumvent the need to culture, then
exactly.
Big hurdle away.
Yeah, or this Streptomyces silicolor M1152. So that actually has its own, or the 4 main natural product clusters of its own actually removed. So when I express my product of interest in that strain, when we do the LCMS, there's no other peaks. It's completely driven towards the expression of your product of interest. It is tailored towards Streptomyces species. So if the gene cluster came from another species of bacteria, it might be a problem. But there definitely are these kind of chassis organisms that we're making that can definitely facilitate that. So there's a lot of promising technology coming forward in terms of new gene clusters and expressing them in other hosts.
That is ridiculously cool. So thank you.
No problem.
It's like sometimes we I don't know, I'm just thinking of that picture where there's like a person breaking out of the prison, but they've made the prison from their own mind, like they've put their own prison up around them, which, some feel like that. It's just in terms of that. Yeah. Well, anyway, thank you so much, Rebecca. We've just gone past 12 o'clock. Thank you so much for your time, and thank you for a really riveting discussion and wonderful.
If anyone has any more questions, you feel free to tweet me or ask me anything else. So yeah, thank you.
Okay, well then, Ryan, I'll hand back to you, and